Now we have another resource person. Many of the students are here from Utkal University. Professor A.B. Das is a professor at Utkal University in the Department of Botany. So we have a long association. Sir, how many years? Over 30 years.
30 years.
Almost 30 years.
More than 30 years.
So we know each other, and good friend of mine. So since he is in Hyderabad, his son is here, and his family is here. So I requested him to give 2 talks, and today and tomorrow. He will be. You all know that for the students, but he is a specialist in plant physiology. He works on various aspects like cytology and molecular cytogenetics, and plant molecular biology, molecular physiology, tissue culture, and genetic information. These are his specialization. I know him as a photosynthesis person, where I was doing my PhD during my JNU days. So they were with Professor Prasanna Mohanty. They are also working on photosynthesis. And he worked a lot on biotic stress and especially salt stress, where he has significantly contributed to study the mangrove physiology. You know the mangroves, right? Mangroves who the plants which grows near to the seawater. So they did a lot of work on a lot of they have identified a lot of biomarkers to identify especially the salt stress. And thank you, sir. Thank you for coming and agreeing to give to the talk.
It is not like
and
that.
You please interact with the students.
It is
So today and tomorrow you'll be here, and please interact with them.
It is my privilege.
Yeah, please.
Because I am not the expert in photosynthesis. Okay, he is the expert. He is the expert.
Let's take this.
We are general biologists. Okay.
Yes.
And in fact, the physiology or molecular physiology, whatever it is there, so that is due to Professor Mohanty. Professor Prasanna Mohanty, he is one of the student. Professor Rajgopal, when he was doing the PhD during that time, I have met him long, and generally used to stay in the JNE guest house. So during that time, I was just imbibed with this physiological part. Earlier, I was a molecular biologist or cytologist, you can say. So that's why today, although the topic is membrane composition, but it is not exactly the membrane composition we will be going for. Maybe tomorrow we will be going for membrane composition in detail. Today we will be going for a general discussion that what exactly it is, what is the chloroplast, and whether whenever we will be talking for this chloroplast, whether it can be engineered or not, and if it is engineered, then what is the differences between the chloroplast engineering and the normal your genetic engineering. So because nowadays a lot of pros and cons are there with regard to the genetic engineering or GMO crop, and that's why perhaps the chloroplast engineering is coming up very fast and giving a lot of what we call emphasis or importance in that particular topics. So in normal cases, because these are the normal slides, I told that I am a teacher of botany. So I am not the molecular biologist or this chloroplast biologist. So most of the cases, if you are looking into that, yes, plastids are, of course, there are the chloroplast, but there are a lot of plastids in the plants, starting from your chromoplast, then chloroplast, leucoplast, amyloplast, etioplast, and so in proteinoplast. So all these plastids, if you are looking to that, yes, of course, there are specialized structures here in the chloroplast, what we are talking about, and Professor Takahashi also talking about, and in a detail. And And I also, I think our students, I also discuss these things quite often, that how you can isolate the chloroplast. So Professor Takahashi was talking about that gradient centrifugation, and of course the sucrose gradient centrifugation. And also I simultaneously asked to my student how you can make a gradient in a centrifuge tube. Most probably they have not answered during that time, and I used to tell that if you are making a gradient centrifugation in a centrifuge tube, say for example 10 to 50%, so how you can make it? So in a test tube, if you are making manually, then how it could be? So what will be the first solutions which can be dropped there in a test tube? Anybody can say? It is a centrifugation of 10% to 50%. In a test tube we are making, or centrifuge tube we are making a gradient of 10, 20, 30, 40, 50. So how you will make that gradient? You just take a capillary tube, put it in the centrifuge tube, put first your 50%, and then 40%, then and so on, so that the lower will be, it will come up, and the bottom portions you will be having the high gradient. And then you can go for a centrifugation so that you can get a separation of the chloroplast. And if you are going to have a chloroplast, say for example isolation, and going for a protein separation with the SDS page, during that time, whether you require a intact chloroplast or it can be a thylakoid membrane. It can be a thylakoid membrane also. So in that cases, can you isolate the thylakoid membrane? Because I think in this workshop they will be going for that how you can make a thylakoid membrane. And initially what Professor Rajgopal was talking about, that we did a lot of work in mangroves, and during that time when we isolate the DNA, during that time we have a lot of difficulties, that in a cytoplasm a lot of phenols are there, and when you are isolating the DNA, during that time the DNA is black color, so all the phenols are there. So that's we started a methodology that isolate the nucleus first, so that take out the cytoplasm, so that all these secondary metabolites it can go out, and then the nuclear palette you can go for the isolation of the DNA. And from that I have at least 2 students, they got the PhD as well as got the job also for this particular isolation process, because one is get the he is a principal scientist in NBRI in cotton. Before that they used to purchase lot of chemicals and what you call your kits from different companies. Then they use this methodology, and one is a principal scientist in Salt Marine Research Institute. So come to the chloroplast and chromoplast, because I'll be going for very quickly, because our sir is talking about that 40 minutes is allowed, it already 15 minutes over, and I have not less than 90 slides. Okay, so chromoplast, because most of the cases you are getting some of the organelles, those are having the pigments synthesis and color, and all these color pigments rather than the chlorophyll, they are, we can say they are the chromoplast, and they are of different sizes. And these chromoplast pigments, the general example is the carrot, where the carotines are much more. And of course the chloroplast and chromoplast, you can find that the green color fruit, they are ripening in the chrome because of the chromoplast, because they are converting this chloroplast into the chromoplast. And of course there are lot of, because most of the cases you have the xanthophylls which is there itself. Can I have some water? and probably these color fruits and all these carrot colors, they are nothing but the xanthophylls, and of course they can be accumulated in the cells like in tomato or in the capsicum. You can get a lot of colors of capsicums in the market, but later on the chloroplast, they become the chromoplast, and that's why the color is different. And also this pheoplast, they are generally found in the phycoxanthin, which is also is a carotenoid pigment, which marks the chlorophyll A, which is the major chlorophyll in the normal photosynthesis process. So where a lot of algae, they have also this phycoxanthin, generally this brown algae, they have as well as diatoms and dinoflagellates, they have this. Rhodoplast is again a phycoerythrin. So this is because of this red color, and there are a lot of red algae which have the same things where we can have pheoxanthin. So all these and the last but not the least, the chloroplast, which is a photosynthesis, and that gives the foods for the millions of peoples in the earth. And of course, we are going to have a 15 days or 20 days of seminar and detailed discussions about the chloroplast. And in these cases, if you are looking into that inner membrane space is about 10 to 12, 20 nanometer wide between the membrane and the chloroplast are small dish-shaped cells, which is nothing but your thylakoid, which is embedded into the stroma. So, and of course, it has DNA because as we believe that this is perhaps evolution-wise, they are the bacterial origin. So that's why they have the DNA, and also the ribosomes similar to those of the photosynthetic bacteria. So as a result, we could see that yes, in the because next day also we'll be interacting with these membrane, what you call protein compositions, and we could see that there are a lot of similarities between these bacterial photosynthetic bacterial membrane compositions as well as the normal hair plant photosynthesis, your chloroplast composition. So as a result, if you are looking to that, yes, of course, we have a stroma and double layer membrane, this chloroplast, and inside the stroma, we have this grana, this stroma are there itself. And can you tell that how these thylakoid membranes, they are stacked, whether it's the inner membrane or it's the outer membrane of this chloroplast? Can you tell anybody? Because the chloroplast is bilayer membrane, so it has an outer membrane, it has an inner membrane also. Whether this inner membrane is pouching to give this type of thylakoid stack, or it is something else. And what about the inner membrane in case of the mitochondria? Because in case of the mitochondria, it is folded and they become the cristae. But in these cases, whether it is an inner membrane or something else. Please interact. Or it is a different structure. No idea. Because we are inside the chloroplast. So that's outside the chloroplast what happens. So we do not know, in fact. But perhaps this thylakoid membrane, because they are not the isolated body. So I think if you are thinking about a cell and the endoplasmic reticulum, whether they have any connections? Endoplasmic reticulum of a cell, how it is connected with a cell? Tell me. Endoplasmic reticulum, it is a folded structure there inside the cell, is it not? So how it is connected with the membrane? Is there any connection with the membrane? Connection. So that's why I am telling the same questions here for the grana, whether these grana are connected with the membrane, so whether it's the inner membrane or outer membrane, that's what I'm asking. Inner membrane. That means you can tell that the inner membrane get invaginated and then become stacked, and that's why it is a thylakoid membrane. Is it? Of course. And if you are looking to the angiosperm, that plant species have about 80% of the plastids are strictly maternally inherited. And that is one of the major features of chloroplast engineering, because maternal inheritance are there itself, there is not any chances of crossing over. So that's why in normal cases, if any gene transfer takes place, it can go for a what you call changes in the next generations, but which is not there itself in these cases, and I will be coming after a few slides. And there are about a particular cell have about 300 plastids in a particular cell, and of course the measurement of each and individual thylakoids is about 2 to 3 micron in diameter, and it consists of large numbers of stacks of thylakoid. And of course, when we did some of the experiment in mangroves, we could see that because mangrove, I do not know whether everybody knows the mangrove or mangrove plants. Yeah, these are halophytes. They are growing in the salt. Yeah. But how these mangrove plants are growing in the salt condition? And why the salt environment is physiologically dry? Because there are plenty of water, but it is physiologically dry. Why? Yeah, that's why. So it cannot take the your normal water. So if it is the case, is how the water is taken by this mangrove plant?
Nematodes.
Yeah, they have nematodes. Yeah. Yes, and of course they have some salt gland. They are excreting the salts from the normal tissue. So in that case, we did some of the experiment in the very high salt tolerant species, and perhaps we published a review paper during 2005 or 4, and that review paper still it is citing, and the citation is more than 6,000. Only one review paper, and that fellow has got the job for that review paper in Sol Marine Research Institute. Okay, so in that case, so as you know that yes, they have these light reactions centers, and of course inside the lumen, of course the light is required for such kind of reactions, and light dependent and light independent reactions are there. And of course, they have the stroma, and about 50% of the protein of the chloroplast, they are found inside the stroma. And of course, the grana, if you are looking into that, they are the normal network of these thylakoid membranes. They are producing inside this what you call matrix, and they are your nothing but your normal stacking, that is your grana, and they may be of 40 to 80 grana in a matrix of a particular chloroplast. So in these cases, as you know that the PS1 and PS2, and of course there are some of the proteins, they are themselves, and also some of these iron and sulfur containing metal proteins are there also, and ATP synthetase and light harvesting complex, they are there. And sometimes also when we teach that light harvesting complex, when we are talking about, and of course the PS1 and PS2. And as you know that the Z scheme, which is mostly modified as well as pioneer in Professor Govindji, he is working for a long time with Professor Govindji, and Professor Prasanna Mahanti, he is no more. But they are, the question is that how this energy is trapped and it is supplied to the PS1 and PS2. Because Sir was talking about the light harvesting complex beside the PS1 or PS2. And this light harvesting complex is nothing but, he also mentioned that it's an antenna. And the antenna size is larger or smaller depending upon the light sources to the chloroplast. If it is a deep-seated, say, your algae, say for example, which is grown about 20 meters from the bottom, from the top of this sea level. So there the light is quite diffuse light. So in that cases, the antenna size will be more or less. What is your opinion? It will be more, because the disk size will be more, so that it can capture large number of light, and that light can be given to the photosystem, so that the photosystem can be operated. And in the same cases, we have seen, I have also worked sometimes in cacti. So cacti, we had a shaded chloroplast. What does it mean? That on the top of the cactus, there is a different color. Inside, we have green color. So as these cactus are grown in a very normal condition, high temperature, high sunlight, so during that time, they have camouflaging this chloroplast, or they are just maintaining the chloroplast in a normal temperature as well as light regime, and top of the chloroplast, they are putting some other color, carotenoids. So these carotenoids are penetrating less amount of your energy, and that is trapped into the chloroplast, so that chloroplast remain intact. So when we did the high amount of your salt, in case of the mangroves, we could see that there are the chloroplast structure or the thylakoid structure, they are get separated out or they are diffused. That means that tightness of thylakoid stacking is generally dismantled. In normal cases, if you are putting high salt or high stress or any high amount of light or even drought or even heavy metals, if you put much more, you will be getting that kind of phenomena. And of course, all these phenomena, when we are talking about that, this, because because it will be covering this lipid part the next day. So if you are looking into the composition of the chloroplast membrane, yes, of course, we have about 35 to 50% of the protein. Lipid, we have about 20 to 30%, and where the fat and sterol and wax and phospholipids are there, and the carbohydrates, also variable carbohydrates, we can get chlorophylls about 9%, carotenoids about 4.5%, nucleic acid and DNA and RNA, we can have there itself inside these chloroplasts. So in that case, if you are looking to the chloroplast and your mitochondria, then we can have a differences of, because the matrix, which is nothing but your stroma here, the thylakoid is your inner membrane of the Christie, and of course the ribosomes are there, and of course the inner membranes and outer membranes both have, and the size wise they are very much smaller as compared to the chloroplast, and of course the origin of this chloroplast and mitochondria, they are nothing but your bacterial origin perhaps. because whatever the molecular evidence what we could get. And energy transductions, if you are looking into that, yes, the chloroplast structures when we will be having that, yes, photosystem 1, photosystem 2, cytochrome B6, B12, so ATP synthetase, and all these protein complex, they are embedded into this membrane. So in that cases, if the membrane is completely intact, and then only you can get all these proteins in a good form, so that the energy transduction can take places and energy is going from one to another, and finally the PS1 can some of the electrons which can be going from this PS1 to PS2, PS2 to PS1 system, or sometimes only one, it is going to the NADPH. So in these cases, all these complex, when these protein complex are there, and of course these lipid bilayer membranes when it is there. So in these cases, these lipid formations are quite essential to hold all these proteins in a harmonic manner. Otherwise, these will be the functions of the electron transfer will be disrupted, so that the photosynthesis, net photosynthesis will be hampered in any cases. So maybe the next day we'll be talking about these bilayer membrane, how it is producing, what are the components of the lipids, and what are the factors which can actually assemble this one. And if you are making some of the mutants, you can see that yes, if you are hampering this structure, or your membrane structure, automatically the stability of these proteins may be hampered, and finally you can have a less your photosynthetic efficiency and PS2 to PS1 efficiency. In these cases, because as I know that you will be getting exposure here for the OGIP analysis here, or you have got some of the OGIP analysis, during that time, that machine can give you the electron transfer system of each and individual protein, your component. So component wise, you are getting a theoretical knowledge that how the electron is moving and where this protein complex is getting damaged. So if any type of external influence, if it is there itself, automatically they can damage any of these part, and automatically the electron flow can be blocked, so that the PS1 may not get that much of electron which is required for the normal photosynthesis. Or if you block this one, only the PS1, if it is operated, so during that time also you will be having a less amount of electron flow, so that the NADPH which is produced there itself, and of course the NADPH is the major things, as well as some of the ATPs which is there itself, which is produced, they are required for the next cycle in the dark reaction. So as a result, what happens that if any of these complex, if it is getting damaged by any of the external factors like the environment or the high temperature, or what you call high salt or high metal, so automatically they can be having the problems. And of course the chlorophyll molecule, those are there itself, because this is the normal structure, as you know, and they have 4 nitrogen bases, and all these nitrogen have the magnesium there itself, and these head portions are nothing but much more of chlorophyll A and chlorophyll B, which is there itself in the head. So if anyhow, if here, and can you imagine this structure, and our we have a hemoglobin structure. So hemoglobin structure also similar here, only the changes here. So it is a Phe. So in that cases, you are, because we are in a virtue of death, only 66. Okay, you are maybe 26 or something like that. So you can think about that, what is the actually evolutionary process of the hemoglobin and this chlorophyll. And my guru, as well as Professor Subramanian's guru, Professor Prasanna Mahanti once told a story that one of the PhD scholar has given a topic that whether the chloroplast can be used as a battery for the pacemaker or not. That is the topic. So if it is, then the solar cell can be implanted in the heart, and so that normally you can move around and they can give the pulses so that the heartbeat can start instead of the pacemaker or the battery, which is generally used there itself, but that was not successful. Okay. But anyhow, you can think about wild things, and of course, a little bit of your chloroplast genomes when we are talking about because a lot of species they have the chloroplast genomes has been done for the sequencing, that starting from Nicotiana tabacum, then Oryza sativa, and so on. And we could see that there are these ribosomes which is 70S, but it is not a eukaryotic ribosome. So it's a prokaryotic rhizome there itself in the chloroplast. So that's why we have also another evidence that might be this is a origin, and also consists of two ribosomal RNAs, the 23S rRNA and 16S rRNA, and also the amino acyl tRNA and amino acyl tRNA synthetase and methyl tRNAs are also found in the chloroplast, and size wise generally about 120 to 217 kilobases. Of course, there are some differences in different organisms, but most of the cases we could see that 95% of this cpDNA in the corn chloroplast has branched and linear size of your DNA. But in normal cases, we have a concept that it is a linear, not a linear structure, it's a circular structure. So instead of that, we could have even in the dinophytes algae, they have a genome which is broken up into about 40 small plasmids, each of 2,000 to 10,000 base pair long. So in that case, a small plasmid like also structures you can have, or a single your circular DNA, or sometimes a linear structure having a branch also you can have. So, and of course, there are major two things, which is the inverted repeats, which is generally hold the genome, if it is a circular one. So where the inverted repeats are there, and all these inverted repeats generally differentiated hold the genomes into small and larger pieces. So one is the LSC and SSC complex, as you know, and about so in this process, if you are going for a genetic engineering for that one, because this is another cases of the GMH, so where we can have a LSC1 and LSC2 and inverted repeats are there. So what will be the proper, which will be the site of your insertion of any of the genes if you are interested to have in the chloroplast? Which will be your site? Because we have shown here that how it is going back. Yeah. So for example, it's LSC1, LSC, and SSC, and we have inverted repeats here, and of course some of the genes which are generally very much common in case of those are very much conserved in nature and which is used for the normal DNA fingerprinting for any of the chloroplast. So my question is that if this is the inverted repeat, this is another inverted repeat which is separated this genome into a large and a small one. So where will be the site of insertion of new genes of your interest? Which will be the portion where it can be inserted? No idea. Okay. think over, and next day you just tell me where can be. Okay. So in these cases, if you are looking into this chloroplast genome, then of course there are about, because in Arabidopsis about 1000 to 1700 generally number of copies, because this is also one of the advantages of the chloroplast engineering, because and in triticum species about 50,000, it is not 1500, 50,000. Okay. And so that's why the chloroplast is unique for the as a transformation tool, because the sequences of the carbon productions and this synthesis of the amino acid, fatty acids, and the pigments, and generally they have the sulfur and nitrogen metabolisms also there itself, and this chromoplast. This pigment synthesis and storage is another factors, and also the leucoplast generally monoterpene synthesis generally occurs there, high protein level and feasibility of expressing of the multiple proteins, and polycystonic mRNA are also found there itself, and there is no any chance of your cross pollination or what you call your F1 or F2 generation segregation. It is not possible there, because no crossing over generally takes places, and of course it's the precursors of the photosynthesis and excellent tools for the your phylogenetic and evolutionary studies. Also lot of evolutionary studies can be done there itself from the chloroplast genome, RFLP analysis of the chloroplast genome. Of course the endosymbiosis is another which is there itself for these cyanobacterial cells as well as engulfed by the heterotrophic eukaryotes, and these photosynthetic bacteria, they have the chloroplast organelles also, so that's single cell. Your chlamydomonas also can be used as a even as a vector. So in that case is also a single chloroplast is there, where also you can target it some of your gene. And in this normal process of the chloroplast generally, we could see that co-expressions of the several transgene operons are generally found there, precise transgene integrations by the homologous recombination. I do not know whether homologous and non-homologous recombination you are aware of or not. Homologous recombination and non-homologous recombination, and feasibility of the expressing of the multiple proteins from the polycystonic mRNA, which is there itself, and regenerations of the crop plants with higher resistance and higher abiotic and biotic stresses for the molecular farming. It is possible to have this one, and also easy process to stack a lot of operons together, and also foreign protein accumulations up to 40% of the total soluble proteins, and in tobacco we could see that about 70%, and absence of position effect due to the lack of compact chromatin structure, and efficient transgene integrations by the homologous recombinations we could find in the chloroplast, and so that if we are comparing this chloroplast transformation and nuclear transformation, we could have, because there are a lot of reservation as well as in our country lot of regulations are there, but till date we do not have much more regulation in the chloroplast transformation, where the reduced gene disposal and maternal inheritance are there, higher expressions and accumulations of the foreign proteins, efficient multiple gene expressions are there, single promoter for the expressions of the multiple subunit complex of the protein, simultaneous expressions of the several genes like the prokaryotic gene expression systems it has, and homologous recombinations which avoid the position effect and gene silencing, and also the transgene is much higher, up to the 18% we could have, which is generally 0.5 to 3% in normal cases. And also here in one thing I could see that that if you are inserting one of the gene into the normal nuclear transformation process, so in that cases if it is a diploid species, then how many positions this gene can be inserted? A single gene you are inserting into a nuclear genome, and a chloroplast genome. So in chloroplast genome, in one position is more than enough to have this expression. But in case of the nuclear genome, so if it is a diploid species, in how many positions at least it should incorporate, if it is expressible? Diploid genome, how many sites? At least 2 sites of a 2 chromosome, at least. Okay, so that the segregation can take place. If it is 1 chromosome, 1 site, then it may not be 50%. Next generation, the 50% will be eliminated, where there is no any transgenic or transgenic information, or in the next generation again 25% will be out. So in that way, after 2-3 generations, everything will be out. So that's why, if that type of things which is not possible here in the chloroplast transformation. Of course, chloroplast transformation is a bit tricky in nature, because isolation of the chloroplast is one of the things. But nowadays, lot of people, they are using the plant and your particle bombardment, where the particle of your tungsten or gold particle, they can be encoded with the plasmid DNA, and then after this particle bombardment can take places, and then regenerations of the plants it can. Or you can use the protoplast fusion type of things, isolate the protoplast, then inject the gene of your interest and into the chloroplast, and then it can be. So in that cases, the chloroplast to regeneration systems you have to develop, otherwise it is not possible or feasible. So that's a lot of examples we could see that, so plasmid, if you are looking to the chromosomes in the plastid genomes as well as the nuclear genome, if you are comparing that, about 60 copies of the single plastid per chromosomes we could have, and genes per chromosomes, a lot of genes are there, but in case of these only very few genes, about 120 to 150. Arrangement of the operon transcription genes, each gene is separate and individually transcribed, whereas many genes are transcribed together. So polycystonic conditions are prevailed here in the plastid system. And we could see that there are a lot of milestones starting from 1988, and we could see that everywhere bialistic, then page bialistic. So page polyethylene glycol, so nothing but the isolation with the chloroplast, then you are fusing this chloroplast, where the Nicotiana tabacum is one of the examples where high level of the foreign protein expressions, and of course the photosynthetic competence, the selection process also, spectromycin or kanamycins, they are generally used for isolation of the your transgene. So which is the selection methods, where in the media itself you can use some of the your spectromycins or kanamycins for the selections process. So in that cases, we should have, because the process of, because from listening itself, we could see that it's a very, very robust technique, or it's a very critical technique. It's not like that. It's only you grow the plant, then after you isolate your DNA of your interest and go for the bialistic. And of course, it is not a target oriented, because every cell cannot be, but some of the cells can get inserted of your gene of interest, and they can go for the plant regeneration system. So where a lot of plants can be regenerated, where some of the plants are transformed in the chloroplast, some are not. So in that cases, because there are a lot of examples where we could see that bialistic and page generally it is used, and where the selection methods are spectromycins and kanamycins, which is there itself, and also because the key conditions for that, the efficient selections of the your transplastomes or your plastids, which is very much important, and a robust method of DNA delivery into the chloroplast, that is very much important to have. And of course, all these things can be done in a aseptic condition, and the presence of active homologous recombination machinery in the plastid, that enhance your capability of the productions of that particular protein of your interest, and ability of the high efficient selections and regeneration protocol, that is very much important to have. At least if you do not have that protocol, then don't go for that type of chloroplast transformation process. And where the gene gun, mostly it is used for that, and generally the leaf cuts or the normal leaves which is grown there in the normal conditions. So then after that these exposed leaf materials, they can be used into the antibiotic conditions, into the media, and or in the biologic conditions also sometimes polyethyl glycol can be used for the treatment of the protoplast for the DNA delivery. And in that case, space technique, generally it is fuse the chloroplast there itself, and also sometimes microsyringe also used, femtosyringe can be used to where the DNA delivery can be done. And sometimes also some of the papers we could see that simply if you are cutting the head of this stigma head and then pour the DNA, then that DNA also can go into the ovule, and that can go for the fertilization process. So in these cases, the microsyringe also can be replaced with the help of such type of techniques in later stages. So there are ample of examples there itself. And gene expressions wise, if you are looking to that, Chlamydomonas and Nicotiana tabacum and Arabidopsis thaliana, so they have a lot of genes which is generally introduced there itself, having the expressions there. And of course, the gene delivery systems, this normal ballistic which is generally used for that, where generally we could have a helium gas where tungsten or gold particles which has been coated with the plasmid DNA, and then after it is bombarded into the tissue in aseptic conditions, and then that tissue can be put into the selective media, and then you can go for the plant regeneration, and these plants can give you a normal plant which can be a transgene plant where the chloroplast can be transformed. So these are the normal handgun which is used for normal leaf where you can also put your DNA. And also the femtosyringe also sometimes instead of nucleus you can put into the chloroplast there itself. And also page methods are also there itself, and there are several advantages and disadvantages appear. Of course, the successful rate is quite low in case of this and random integration of the foreign DNA into the genome, which may be a unstable, produce an unstable track. And list of transcriptomics plants which is generally found where a lot of genes has been already introduced and get the successful result. Of course, there are some your LIC genes or GFP genes, they are normal genes which is generally inserted there itself and got the results there. And say for example, TIG40 his genes, Arabidopsis thaliana, it has been incorporated there itself in the chloroplast, or in the GMAge, the mannose 6-phosphate isomerase also has been introduced. So in that cases, and these are the normal process of your chloroplast transformation process. This is the theoretical aspects where we have to have a gene of interest, and of course we will be having a selectable markers, and also the plastid expressions. Some of the expression genes also has to be incorporated here, and insertion site regulatory sequences we have to put there, and of course some of the reporter genes also you have to have so that you can get informations that whether this product can be inserted there or not, and it can be validated with the help of the PCR by or the southern blotting technique with the raising of primers, and in that process there are a lot of applications starting from your metabolic pathway engineering, research on DNA, RNA editing, then phytoremediation, products of industrial enzymes and biofuels also can be produced here. There are a lot of ample of examples where also a lot of genes site of integrations, regulatory sequences, and transgenes which have been also there itself, and what is the effect as well as efficiency of these genes which is there itself we could see, and also the pharmaceutical and vaccines in the plants also can be targeted there itself, and phytoremediations also sometimes mercury, because mercury and reactase enzymes has been bacterial genes which has been inserted into the chloroplast, and they are accumulating that your mercury from the environment or from the your substrate. So, and also some of these enzymes like hydroxybenzoic acid, this PHBA, that also can be also produced, and also some of these PHB also it can be also produced there itself in the normal plant where it can be there itself. So as a whole, if you are looking to that, of course you can target a chloroplast where gene of interest can be inserted, and then selective media you have to go for, and theoretically it is nothing but you targeting a single chloroplast, then this chloroplast is multiplied there in the next generations. So where cell divisions can be produced there itself, and of course this transgene they can be put it into a selective media, and then after growing of the new plants it can be there, and testing of these plants whether it is a transgene or not, and of course if it is a chloroplast oriented, automatically the it has several advantages over the nuclear genome insertion. So as a result you can have a uniform and homeoplastic conditions of your normal transgenes which is there itself, and of course there are a lot of copies of these particular genome are there in a particular chloroplast, so that it can produce a large number of the proteins instead of a single protein. So copy number is very much high there itself. So in this process generally the when you are putting you're preparing a vector, so in that cases normal cases we have to have a LTR sequences, because as I told that where your insertions can be, so LTR and you have a RTR, so inside this you can put sequences there. So there are some your sequences of UTR region as well as 5' end to 3' end, and where the gene of interest are there itself, and there all the genes can be inserted here itself, because NPT2 they are the marker genes, or BADH they are marker genes there. So gene specific activator generally we can put there itself, and so that it can active the promoter. So promoter, then activator, and some of the UTR region, then promoter, then activator, then you have your gene of interest and UTR system, so that you can insert all these construct into the your transgene, and so that finally you can have the expressions of that inside the chloroplast. So all these transgenes it can be targeted into a chloroplast, and then this chloroplast can be multiplied there itself into the system, and they can go for the expression, so that after the transformations you can have a transgene expressions, and it has a lot of advantages as compared to the normal genome sequences. And I am just completed. It is one, and if you have some questions, it's a very wide and general topic. If you are welcome for any of the questions. Thank you very much. Any question? Or yeah, please. Yeah, PhD engineering already, because I have given this one, and it is there along with your system. You can take, because I have just skip very quickly. But PhD you can have, because in the synocystis generally PhD preparations generally it can. So that is there. Phytoremediation, mercury, already it has been established. Anything?
Sir, when we're talking about the orientation of the photosynthesis component in the orientation of the photosynthesis component in the thylakoid membrane, it is like PS2, cytochrome P6F, and PS1 and ATPase synthesized. But there is any different orientation or always in same orientation?
As far my knowledge goes, it is the same orientation.
Okay. The previous slide I saw like something else.
Is it like that? I don't know. PS2 to PS1 generally. This one? Where it is just take a look. Yeah, just yeah, tell me. Yeah. Yeah, tell me.
Yes.
Yeah. PS1.
PS2 is white.
PS2 is white. Yeah. Orientation. This photograph we can orient this way. Is it not? This photograph, if it is upside down, yeah, yeah, PS1, PS2, if you are upside down this way, just reverse, then it will be your answer. Is it not? You just upside this one, this photograph is just upside down.
Okay.
Then it will be your first one, will be your 2 and then 1. Because the next slide is 2 and 1. The Z scheme generally 2 to 1. Anything? But good thing, at least you have pointed out that one minutely. Good. Anything?
Sir will be here for more time. Thank you, sir. Thank you for.
Oh, thank you very much. Thank you very much for everyone. Yeah, sometimes actually, very frankly speaking, I am going for a lot of interviews for the biotechnology student. And even I ask whether the rice is monochrote or diploid, I am not getting the good answer. So that happens sometimes. When we are entering into a system, we are forgetting the outer system. The outer system you should keep in your mind what exactly it is going on. Then the inner side, it's of course the detailed things, it is very much important for your PhD program. But after the PhD program, you will be going for job. So during that time, you have to correlate the inner thing and outer thing, the wide thing, because if I am a bureaucrat there, sometimes you will be getting a lot of bureaucrats in the interview board. They will be asking very general question, and it is out of your syllabus even. So that's why you have to be you have to cross the bumpy road so that you can tackle very well. Just I have tackled the question of this gentleman. Okay, thank you very much. Thank you very much. Yeah, thank you very much.